Okay, great. Thanks a lot, Harmon. So now, given that we have, there's nobody speaking after me, right? Okay, all right. So the bottom line is you all can ask any questions, stop along the way, etc. And I have a decent amount of material here to go through, but I'm going to do it at a fairly leisurely pace. And as Harmon mentioned, I will actually sit up at kind of a high level and then dive in. And I'm also going to give you a lot of kind of running commentary on things. But I'm going to divide what I'm going to talk to you about in three areas. So the first part, I'm just going to talk about general properties of regulatory DNA, and I'll define regulatory DNA in just a minute. And this is something that we've been interested in and studying now for many years. But I want to just kind of recap for you what I think are the main properties that have emerged about the regulatory DNA compartment that we know so far, and some of these properties coming out fairly recently. But that these are things that you sort of need to keep in mind when you're kind of approaching any kind of analyses of regulatory DNA. And in the next parts, I'm going to talk about actually some work which is largely unpublished, but which shows sort of carry forward of some of these principles into a couple of biological settings. One of them is human fetal development, and I'm going to talk about not only regulatory DNA, but also transcription during early human development. And there are also some, new surprising things that come out of that in terms of general principles of how to think about regulatory DNA and regulation in general. And then I'm also then gonna sort of wrap up by talking about kind of this question of where did it all come from? Why do we have all this regulatory DNA? And I think there's a lot of, you can call it confusion, you can call it conflicting theories, you can call it conflicting, perspectives, whatever, on the role of sort of evolution and conservation and our interpretation of what's going on in the regulatory DNA compartment. And so I want to talk about that and try to give you a framework for thinking about how you approach that. So to start out with, I just want to, just give a brief 1, 2, 3 about something we'll call the living genome here. And this is something, of course, probably everybody in this room knows. DNA is packaged up into chromatin. It's wound into these higher order structures that we don't really know a heck of a lot about. Somehow it's packaged all into the nucleus. The genes obviously are packaged in here. And then there are these other kind of landmark entities in the genome, which are regions where the repeating unit of chromatin is interrupted by short stretches, a couple of hundred base pairs, where sequence-specific DNA binding proteins are complexed and typically binding in a cooperative fashion that gets rid of or at least outcompetes a nucleosome in these positions. And we know now, I'm generically calling this stretch of DNA that encodes this activity regulatory DNA. And the reason for that is that, which will become clear in a little bit, is that we know every time we can track down certain properties that relate to biomechanical events in the genome that at least are DNA encoded, they tend to end up in one or two compartments. They're going to end up in the protein coding compartment, or they're going to end up in this in this compartment here. But the thing is that our perspective currently on exactly what these elements are doing is fairly limited, and I'll talk about that in a minute. So, within the regulatory DNA, as I mentioned a minute ago, you have these proteins. Now, we colloquially refer to these as transcription factors, but you should be aware that that term encompasses not only some sequence-specific DNA binding proteins, but a lot of other activities which don't touch DNA, which are involved in the transcription process. But usually when somebody says the word transcription factor, they're meaning proteins which are DNA binding proteins. And of course, each one of these guys, recognizes a particular set of, well, it recognizes something. And what it recognizes is not the DNA sequence. Okay, so that's kind of an important thing to keep in mind. What it is recognizing actually is the shape of the DNA. So most of these proteins are not contacting the Watson-Crick base pairs. They're essentially binding in one of the major or minor grooves of DNA, and they're recognizing a particular shape. And so every little transition between bases in DNA causes some deviation in the pitch and the roll, which affects the shape of those groups. And that's really what the factors are recognizing. And we read that out, though, as a surrogate with certain sequences. And again, if you look at I'm sure you guys have covered motifs and everything so far, and there are various ways to look at this. But that's really, again, it's a surrogate measure for sort of what the proteins are recognizing. And then, of course, what happens in here is that when these proteins bind, they have certain components of them that are their DNA binding domains, and they have all these other things out there in the protein that are largely mysterious. And you know, if you're a student and you want to work on something incredibly important that nobody works on, you can work on what it is about all these other domains that allow them to do things like recruit in activities that put on histone tail modifications in all kinds of different combinations, et cetera. And I'm not going to go into any further discussion of histone modifications, DNA methylation, and sort of other epigenomic type features, except to say one thing, and that is that that basically the histone modifications occur on nucleosomes which are by definition around the regulatory DNA. They are not in the regulatory DNA. So always when you look at high resolution where that signal is coming from, when you look at histone modification signal, it's always in the regions that are immediately flanking the regulatory DNA. And of course it may spread out from there a little bit. The second one is with relationship to DNA methylation. And here there is actually a fairly simple relationship, which is that basically, when the transcription factors are here, these templates are essentially constitutively unmethylated with just some, rare counterexamples. So essentially, these are completely dichotomous compartments. When the factors are there, there's no methylation. But when the factors go away, methylation seems to come in kind of a quantitative way, which suggests that it's sort of a passive mechanism that's kind of chasing the transcription factors rather than kind of being actively put on there. So, so I mentioned, the idea of this term, regulatory DNA, and we have other names for these kinds of regions. You call them promoters, enhancers, silencers, insulators, and I put the et cetera. But the problem is that the list stops right about there. And this essentially, these terms are operationally derived terms. They were derived out of assays that people developed in the 1980s, in the early half of the 80s. In the first three cases, to describe a phenomenology that they were seeing. And the problem with this is that imagine if you went to school and you learned exactly 4 words in kindergarten, and that was it. Now you had to use these words to describe everything going forward. You would rapidly run into this big problem that the vocabulary was not sufficient to account for your experience. And so this is kind of the problem that we face at the moment, is that we sort of know that there are these kind of basic operationally defined activities, but in reality, we don't know a whole lot about really what are the categories that are out there. Well, we can probably be pretty sure that not all these elements fall into these little categories, but the problem with this, which is a very dangerous problem, which is continuously re-emerging now in the literature, is that there is a general tendency to say, oh, if it's here, it's a promoter. If it's out somewhere else, it must be an enhancer. And you see this on and on and on again, and this is very, I think, kind of insidious thinking that imposes mechanistic views on elements that are out there. And I'm going to comment about that a little bit later and actually put some numbers on some of these things. Yeah. Well, I guess by definition, promoters are not distal. But do you mean, do you find new promoters that are sitting out there? Oh, there's tons. So if you look, at the promoter landscape, and if you look in like the, if you look at an annotation that's based on transcription initiation, like the GenCode annotation, you will find that there are a huge number of, regions upstream of transcription initiation, which you might call a promoter. Now, many of those are only offset from each other by a few bases, so it's not really a promoter. A promoter, we know, is an operational unit, and it has a certain set of chromatin features. We know there are transcription factors there, we know, et cetera. So if you call that kind of the biological unit, and then you resolve that with the GenCode transcripts, you end up with a number that there's somewhere around, if you pull in all of the ENCODE data, in other words, what things do we have biological sort of evidence for, there's somewhere in the order of about, I don't know if Anshul has any other calculations, but we calculated this relatively recently, somewhere in the range of about 170, 180,000 of these guys that are out there. So it's not a huge, I mean, it sounds large, right? But a lot of these are, so of those, maybe, you know, I would say 50,000 might be what we call canonical promoters, and that's about 20,000 coding and about 30,000 roughly of these sort of well-structured non-coding genes. And then all of those have alternative promoters. So that means that the average gene out there has maybe, you know, 3 or 4, whatever, alternative promoters. But
so when you say alternative promoter, do you mean like one case where it could just be a few bases?
No, these are all, and when I mean alternative, it means that you have moved into the range of a different biological entity. In other words, so if the promoter is, let's say there's a 500 base pair piece, let's pick it where the transcription factors all are binding, there's nucleosomes on either side, okay? That's one unit. By definition, the next one of these units can't occur until the next sort of nucleosome over. That's like the closest it could possibly be, okay? Yes, but they're typically located farther away, and it also depends on the size of the gene. And within that zone, there is, of a promoter, there's all kinds of different spots where transcription may initiate, depending on certain circumstances. There seems to be sort of a canonical region around it which coalesces. So you have to distinguish between alternative initiation that's related to the same biological promoter from different sort of biological. And the thing about it is that these alternative promoters are largely, they're much more tissue specific than, or selective than the main promoters. But in general, as I'll mention in a minute, that promoters in general are not very selective. So you see them in lots and lots of cell types. And so they're really integrators of information. They're not really, providing the information that's specifying, cell specificity programs, essentially.
But if it was tissue specific, you would think that perhaps the 5' UTR might differentially regulate that result in transcript in a different manner with the other alternative promoter.
Well, I mean, if you find one that's completely, I mean, here's the thing, is that the UTR also, there's a lot of information in the UTR as well. So there's, typically of a promoter, but sometimes the UTR is also, that information is also separated from the promoter by a nucleosome. Sometimes it actually encroaches on it. It really depends on the gene. I'm going to get a little bit later to the topic of sort of where the information is in that sphere. But I think the basic idea is that there's still a limit, as I'll cover here just in a minute, that there's some limited amount of space that you might call promoter space. There's a canonical space, there's the alternative space, and in terms of biological units, that's about how big that the spaces are. So we have been engaged in trying to define essentially where all these regions are in the genome. This is something that started out, I mean, there have been a variety of different technologies, really a succession of them thrown at this problem for the last decade. Now and various other ones have gained traction, particularly sequencing-based approaches. And what we have done is to leverage a molecule which has been around forever, and it really was the major modality for identifying regulatory DNA in the pre-genome era, which was DNase-1. And so DNase-1, basically, when you drop this guy into the nucleus, this was observed originally, so this whole idea was brought about by Harold Weintraub and Mark Grudin in the 1970s. They had a very famous paper in 1976 in Science demonstrating the basic concept that if you put in DNase-1 in a nucleus, essentially cleaves kind of active genes, and it was really actually an amazing paper. It was done in kind of an era even before Southern blots were invented, and they had all these super clever assays for things. And then around 1980, there were several different groups, Carl Wu and Sally Elgin, Gary Felsenfeld, and Weintraub again, that came back and noticed that in fact what's going on with DNase-1 is if you take a high resolution look, because in the intervening years they had Southern blots that had come around, they were able to say, well, gee, actually these cleavages are super localized in the genome. They localize over small stretches of DNA, and they create these regions called DNase hypersensitive sites. There were sort of a succession of papers around this, and then people kind of went bananas with the method, using it in the old-fashioned way, just doing one southern blot at 4 kb at a time or whatever, to try to find regions. Weintraub was the first one actually to map these, because everybody thought, well, everything's at the promoter, and then Weintraub actually was the one that discovered, no, in fact, these things are all over in and around genes. There are these distal elements that are out there. And then this actually led to, or merged with some work with others to define the first cellular enhancers, et cetera. But basically what happens, and what we now recognize what's going on when you drop DNase 1 in the nucleus, is that it kind of vaporizes the sequence that's in here, and it releases a bunch of little fragments. And so what it's doing is it's actually, as I'll comment on a little bit later, it's kind of cutting in between the proteins, and it may maybe put one cut here and one cut there, so you can have cuts that start in here and end on a nucleosome, cuts that go in between different proteins, et cetera. So it releases a range of these little DNA fragments, and then what you can do is you can just collect these guys and you sequence them. And the thing about them is that all of them, by definition, have DNase cleavages on both ends. Okay, so these are what we call the double hit fragments. And it turns out that these fragments basically don't come out of anywhere in the genome except for these DNase hypersensitive sites. So what you can do then is you collect these guys, you sequence them, you map the reads to the genome, and then what you're doing then is creating effectively a genome-wide map of where all the cleavages were that the DNase 1 saw. And so and then what you can do is then you can go and you can tally those as you move across. So let's say you did this for a couple of different cell types, and we're looking at about 100 kilobases here. And then you can just go across and count the cleavages. Now what graphs like this show, which is the typical thing you see when you pull open a genome browser, is that somebody's taken a roughly nucleosome size window, about 150 base pair window, and you're sliding it across the genome in about 20 base pair steps, just for convenience, and just tallying at every point. How many cleavages do I see? How many do I see? And so, sliding across, you get these kinds of pictures of peaks, and these are the, these guys up here, the DNase hypersensitive sites. And really, what we're talking about is that the tip of these peaks is where all the, all the action is going to be. And then on a more generic level, then, these peaks are sort of marking where the regulatory DNA compartment is without necessarily telling you anything about what it is that all these elements are doing. So these are pretty much something that we know that there are factors that are binding in the genome. And just a couple of comments on this. It turns out that these sequences, or these elements, really only result from this factor binding. So in other words, it doesn't appear to be any place in the genome that DNase 1 can create these kind of hypersensitive sites in the absence of factors being there. So if you kind of do this across many different cell types, and this is just a tiny snapshot of how many there are really available now, you get these maps. And again, as I mentioned, there's the hypersensitive sites, and they kind of mark up all the promoters, and they also mark up the enhancers, or well, here I'm calling this happen to be enhancers, but let's call them the distal elements generally. But if you went around the genome and you tallied up how many of these peaks there are, and you just counted them up in any given cell type, what you find that there's somewhere between about 100,000 and 250,000 of these guys per cell type. So that's roughly about 1% of the genome on average. Now, a couple of qualifications here. When these numbers are based on identifying these peaks using a fairly straightforward approach and also imposes a false discovery rate threshold of about 1% on that. So at the false discovery rate 1% level, these are sort of the numbers that you get out there. And again, these numbers are going to become important a little bit later on because it turns out about their distribution, different cell types, et cetera. But anyway, in any given cell type, small compartment, think about 1% of the genome, because each of these elements on average, those peaks is around 150 or so base pairs. So, but the other thing about this compartment is that there's, you can see there's a lot of variation between cell types, and so to get kind of a complete map of everything, you need to go sample lots and lots of cell types. And this is a figure actually out of the roadmap epigenomics papers, but I just put it up because it has lots of cell types and things on it that have been sampled by DNase 1. You know, so really there has been a fairly broad effort, and I think that now there are DNase 1 data sets for over 400 different cell tissue types and states, meaning the same tissue, maybe at different you know, times of development or something like that. So there's quite a lot that are out there. And what that means then is that you can kind of go to any place in the genome now that you want. You can put your magnifying glass on it and you can pull up any number of cell types that you want. And you can see the regulatory DNA kind of flipping on and off. And these are fairly big regions here, a couple hundred kV or so, I think in either case. But it also kind of gives you a picture that there is really fairly stunning cell selectivity out there. So here's, in this cell type, you know, there's tons of elements. This is a globin locus, they're just totally, you know, all over, they're on. And here you go down to, like, you know, these B cells, and it's just completely flat as a pancake. There's nothing there. So there's a lot of really kind of black and white activity in this compartment. And what that means also is that there is, you know, a lot of cell and state selective elements. And in fact, there is an incredible lot of these elements we now recognize. So that if you sort of go around and you kind of make a tally of where things stand in the genome, and this will get back to one of the questions earlier. So if you look, you know, in the gene compartment, let's say, so there's roughly about 55,000 of these by one count. There's about 21,000, roughly, 20,700 well-defined protein coding genes. Is again this number here, this long non-coding RNA gene number. This bounces around a little bit, but I think that there's, again, these are the ones that are sort of well-defined. They've got a promoter, you can see them, they're spliced, they have other features. And of course, there's just a ton of smaller non-coding RNAs, many of which are highly reproducibly produced from specific spots in the genome, but we don't really have any way to categorize them into a framework that we kind of understand as genes. And not all of these, for example, have an adjoining promoter or anything like that that allows us to kind of relate them to the framework of genes as we understand it. All right, well, that's that compartment. And then I was mentioning also that there are alternative, you know, so into this 55,000, there are somewhere about 180,000 promoters that are in there. But when you look at this compartment here, there seem to be, at the moment, we're now well over 3 million distinct entities that are out there that have been seen reproducibly in a particular cell or tissue type or state, and they're often in others. I mean, most of these are extremely compartmentalized in different cells and tissues. And it seems like a lot of information, but remember, only somewhere between 100 and 200,000 or so of these are on in any given cell type. So there's tremendous combinatorial usage, or really sort of sub-usage of this really big compartment that's out there. And so I think it also means that the information, this regulatory information, is incredibly densely encoded in the genome, and you know, it's just actualized in a very selective way. And then if you ask at the level of the DNA binding proteins, you know, we currently think that there's somewhere north of about 20 million spots on the genome that encode a recognition sequence for a DNA binding protein. So these are kind of our current numbers, and they're probably only going to go up. And you know, the question I mentioned earlier is, you know, what do all these elements do? And you know, we talked about the fact that we sort of have this kindergarten vocabulary for describing this. But really now, what is emerging by putting together lots of the information, types of information that have become available through projects like the ENCODE and Roadmap, et cetera, what we realize is that there are elements out there. It's not like every element has its own function. There are groups of elements that seem to have common functions, but these functions are very complex and really can only be realized once you start to put a lot of information together. So for example, you know, last summer we had described this, that there's a class of about, you know, 10 or 12,000 elements out there, probably more actually, but just in the set we looked at, that are these marked by DNase hypersensitive sites that are sort of parked, you know, next to and encroaching on exons. And what these things do is, across a wide variety of genes, is they seem to be involved in looping the gene or the exon over to the promoter region, where it's kind of encountering the co-transcriptional splicing complex, and that's modifying the rate of alternative splicing of these exons in a tissue-specific fashion. So if you see the site show up in tissue X, then what you see is you see the connection that is being looped over, which you can map by chromatin interactions, and then you can see this effect on splicing. So again, there's this cohort of elements that seem to appear, you know, to behave in roughly the same way, but we don't really have any kind of name for these yet, and undoubtedly there are many, many, many other categories like this which have yet to be defined. But I think one of the other sort of problems is that there's nobody actually sitting down and defining all these categories. You know, there's no body out there that is really making a very concerted effort to put the categories all together. And of course, this is an incredibly daunting task to do it, and you need a lot of data. And you know, because most of the efforts out there now are just focused on kind of finding everything to begin with. So you know, there's also the fact that regulatory DNA, you know, even in the simplest circumstance of, let's say, a distal enhancer or something, is interacting with some kind of target gene. And that interaction has many interesting properties. And it was something that was recognized a long time ago, obviously, that there was distal control, and it was also recognized that this relationship didn't happen by, you know, teleportation. It happened by physical approximation of these elements with their target promoter. Now, there is a natural tendency to think that the activity of this, so in other words, I have an enhancer, the enhancer is controlling a promoter, and the promoter is transcribing stuff, that if there's physical approximation involved, that this output is dependent on this thing touching this. Okay, this is like the natural thing people think of. It's like, oh, you know, the interaction should be there, and I see something. But the thing about it is, what that is assuming is that this interaction is a trophic interaction. In other words, that it has to sort of be going on like this in order for the RNA to come out. And there used to be two views. So this actually comes down to a really fundamental question about the mechanism by which distal elements, we call them enhancers, whatever, that affect transcription are working. And there were two colliding and competing schools of thought around this in the 1980s. And it came to a head around 1986 with a couple of papers from Tom Maniotis and also from Walter Schaffner that were published in Nature that suggested that what enhancers are doing is to increase the rate of transcription of a gene. So that they were basically like a dial that you turned up when you had it there, and the gene would produce more RNA. And what this implied is that the enhancer is actually there sort of doing it and making the whole thing happen. And they presented a set of data for this, and then subsequently Harold Weintraub took a look at this and said, you know, there's a completely different explanation for this. And the different explanation, which he went on to provide some extremely compelling data for in a couple of beautiful publications. And the alternative explanation is that, in fact, what this thing is doing is, remember, when we do any kind of assays, any of these high-throughput assays you've been hearing about, they're generally all done on populations of cells. And so what he came around to is the idea that what these elements are doing is that they're not increasing the rate of transcription. What they're doing is they're acting kind of like an enzyme. And what they're doing is they're catalyzing the formation of an active transcription complex on a piece of chromatin. So that if you have a population of 500,000 cells, you have 1 million templates, right? They're diploid cells. And that what the enhancer is doing is just increasing the probability that this thing will just start happening on any given template. And that when that thing pops into that state, then it stays there and it's kind of active, okay? And so this means, under this kind of model, which I think has more evidence behind it, this guy over here doesn't need to be touching the enhancer all the time to do it. It's sort of like those, vaudeville things where they set up the plate on the stick spinning, right? And they set it up, and the guy goes by occasionally and gives it a hit here and there. But by and large, it's moving. And so it makes the whole thing, the whole picture, a little bit more difficult to disentangle, right? You can't just go and just kind of measure all the interactions and immediately kind of trace everything through. But we certainly have come to recognize that the genome is pretty tremendously interconnected in cis, meaning sort of along the chromosomes. And one way that you can see this is because most of these elements are cell type selective. If you have 10 cell types, you don't really get a lot of information. If you've got 100 cell types, then you can start looking across the genome at, let's say, a given promoter. And you can say, well, when this promoter is on, what other things are on around it within, let's say, 100 or 500 kilobases, okay? And it turns out that you can see these really stunning patterns that, the promoters flip on, and then suddenly, and with them, all these other elements, when the promoter's off, all these other elements go off. And they keep doing it in this pattern, over many different cell types, both on and off. And then what you can do is you can confidently say, start to make statistical associations between this distal element and this, and this promoter. And it turns out that when you do that, you basically come up with certain interesting numbers. And I should mention that when you see elements that are highly correlated like that, their behavior is tightly linked, their activity is tightly linked to a promoter, that those elements, when you go back and then measure the frequency of physical interactions, you almost invariably find for the highly correlated elements, those that have correlation coefficients above around 0.7, you invariably find that there are physical interactions. So it seems that those spaces kind of converge. And some of the basic numbers that you get out of it. So if you kind of crank across those hundreds of cell types, you set a high threshold at which we know that the correlations are matching up with physical interactions, and you come out, again, with about 800,000 elements that you can assign to their cognate gene. And if you do some statistics, or just some basic counts around this, what this kind of means, and when you tally it up, is that the genome looks quite different depending on where you're standing. So if you're standing on the promoter, it looks like the average promoter is receiving input from around 20 distal elements. It doesn't mean that it's getting them all at the same time. It means across its kind of life cycle of being used in many different cell and tissue types that there are sort of 20 elements on average that are feeding in. Now, of course, some have more, some have less. But if you then shift your perspective and stand over here in the genome, then things look quite different, because it turns out here that half of those distal elements, that huge compartment out there, seem to be connected to a single gene. Right? That is what they're after, and they're not really having other roles. And then there's another roughly about 30% that interestingly connect up with two genes, and again, not necessarily always in the same cell type. So it could be this gene in this cell type, this gene in another cell type, and there's around 20% that regulate 3 or more genes. But it turns out that the lion's share of these appear with genes that occur in clusters like cytokines, et cetera, which are paralogous duplication events out there. Or that example like with the globin locus that I showed you, which is a paralogous locus. So in other words, it's sort of coordinated gene systems. So those are the basic numbers. But then what you can do is you could say, okay, if I go and I look at the sequence in the promoter, and I look at the sequence in this distal element, which is connected with this, supposedly connected with this promoter, are there any common features? Like if I see this here, do I see this here? And it turns out that there are some really stunning relationships here that fall out. So that you can find, for example, that when you look in this framework, that there are certain particular sets of recognition sequences for factors that you find in distal elements and their cognate promoters, but not in, let's say, some other promoter. So that there appear, in other words, to be some type of pairing rules that are reflected in the sequence that relate to the sort of target to, whatever, the predator to its prey, let's say, in terms of these connections. And then you find some really interesting things. So if you look at, let's say, pluripotency factors, so these are things like NANOG and OCT4 and SOX2, these kind of famous things, KLF4, Yamanaka factors. Well, if you look at, let's say, in ES cells, and you look at things that you find in distal elements that are connected to a particular promoter, you find that promoters that have NANOG have distal elements that have SOX2 and KLF4, but not NANOG or OCT4. The most extreme example is that if you have a promoter that has KLF4 it is only connected to distal elements that also contain KLF4 and never any of these other factors. So there's all these rules that are there and are really kind of begging exploration. So what I want to do now, and just I've kind of painted some very general things, and what I want to do now is turn to talk about some general biological properties of the regulatory DNA compartment that we've come to recognize. And the first one of these is, and I would call this probably the cardinal property of regulatory DNA, is that the actuation of these elements is really extremely cell type selective. And again, back to this image I showed you earlier, what it means is essentially is that most of the genome looks like this, with this kind of behavior, and not like this, which is the kind of thing that you see around promoters. And so, by and large, again, these distal elements are highly selective. They're only on in a few percent of those hundreds of different cells on average. There are some elements, of course, that defy the trend that are on in more, but the vast majority of them are extremely selective. On the promoter side, you find that, again, most of these elements are non-selective, although there are a few out there that are selective, and they tend to be the alternative promoters. But again, the promoters are maybe 5% of the landscape in terms of the regulatory DNA.
Would you say actuation? Do you mean active?
I mean that they are that you can detect that that the element has been primed, at least, okay? Because the phenomenon we're just detecting is some factors have shown up in this particular cell type in a cooperative manner. They've punted the nucleosome, and they're there. Now, we know that phenomenon can precede certain activation, but it is necessary. So I'm thinking of that the way you think about this is kind of like starting the car and driving the car, okay? And so these are the ones where this car has been started, at least.
So there's transcription factors that are bound, that are present, but whether or not they're active has not been demonstrated.
Yeah, what they're doing is in other words, that the element is either, the old view of this is the element was primed for activation. So many times what would happen is these elements would turn on, almost invariably, elements that were turning on if you looked at in a differentiation series before they were actually sort of utilized, okay? But as I'll talk about a little bit later, it turns out that we've just recently recognized that there's a whole other compartment of things that get turned on, they're used, and then instead of shutting off, they actually stay on. And they provide a memory of an event. But I'll get to those in a little bit. So another really important property is that the information here is in these regions is very, very densely encoded. And in each of these cases, when you look in, these regions are essentially densely populated by transcription factors. Which means that in any one of these regions, there are the regions that are actually, or the bases that are actually contacting the factor, and there are bases that are between the factors. And one of the remarkable things about it is that there's two kinds of information here. One that is sort of structural information that is immediately recognized, and then the second one is information that is in the molecule that may affect the recognition of a protein nearby. So, you can go and you can tweak the DNA in between where the proteins bind, and it actually can affect the way that they bind, with each other. So we tend to think of these as an entire unit that has these other components in there, but it's clearly behaving as a unit because it has this, evil nemes, which is the nucleosome that it's competing with. And we essentially have the nucleosomes to thank for everything because without nucleosomes there would be no cooperativity, and without no cooperativity there would be no cell-specific gene regulation, and we wouldn't be here. Okay, so what does this look like when you look at transcription factors here on a one-by-one basis using a method like ChIP-seq? So you can go, for example, to a region, a couple different regions here. So this is a region about 50 kilobase or so, yes, around chromosome 19. This is done in one of the encode reference cell types in K562. Here's the DNase 1 profile in red. Here's another region, a little smaller, 10 kb. This is around this beta globin locus control region. And what I have down here are the traces from the ChIP-seq profiles of, I don't know, about 40, 50 different factors from ENCODE. And I just put as many fit on the screen, right? You can keep going with this. And then what you can basically see then is that the, that these obviously all have their own little nice peaky profiles. But if you take all this information here and you just literally, normalize it for the number of reads, and you just add it together, you get this track right here, this black track, okay? And what that basically shows you is a couple of things. First of all, on a qualitative basis, meaning kind of yes, no along the genome, this signal is basically concentrating, in the signal where the DNA sites are, and we're not having any appreciable signal that's outside of there. And the second thing is that there's also a quantitative relationship here too, right? You see how these peak heights kind of track, with the combined density of the transcription factors as well. Now, that phenomenon has a much more complicated explanation. Because the thing you're thinking is, oh, that's great, that this is just kind of a generic readout for, the concerted activity of all these factors. The problem with this view is that we know, so for example, this right here is easily the single best studied regulatory element of any organism on the planet. And this region right here, the beta globin locus control region, I think at last count has about 1,000 papers on it, with well over 100,000 citations. It has had every permutation of experiment you could imagine done on it, and a lot of them have been done on this guy right here. And we know that that element has a finite number, has about 6 different factors that are in there and touching the genome. We know they've been mutated ad nauseum and studied in all kinds of contexts, so we know what lives there. However, what you see in the ChIP-seq signal is a lot of signal here, right? And naively you would say, oh, well, there's really 20 factors there. Well, I think that the basic answer is there's not really 20 factors that are there that are touching the genome. There are a limited number of factors that are there touching the genome, and then there are a lot of other factors that are touching a lot of other factors that are eventually touching something that's touching the genome, or they just happen to be hanging out nearby when the cross-linking happened. And then by extension, this is probably the case over here, because again, there's a finite amount of real estate. You can only pack so many proteins into a 150 base pair region, and yet you see these signals. And so with the ChIP-seq data in general, I'm not going to go into this any further, but I'll just give you a general perspective on this. When you do ChIP-seq and it involves cross-linking, so the vast majority of data that are out there involve formaldehyde cross-linking, you get a lot of extra stuff. Because remember, let's say when you're mapping chromatin interactions, you're also using cross-linking. And so you've got basically the same sample can give you 2 kinds of information. It can give you occupancy information, and it can give you proximity information. And you can't necessarily readily separate them. So it's the case. So what happens then is that each of these signals that you see here is a combination of some amount of signal that's deriving from direct occupancy of the DNA, and some amount of signal that is arising from the proximity to other factors which may be directly occupying the DNA. And this is also reflected when you look in the motif content. So if you take transcription factors, let's say you take all the transcription factors from ENCODE, and you look and ask what proportion of the peaks have the cognate recognition sequence of the transcription factor. Now that number ranges from about 5% to about 80%, 80 or so percent. And with most factors hovering in the middle of around 50 or so percent. So in other words, that you're getting essentially a composite signal when you're looking at ChIP-seq, at least when it's done with cross-linking. It seems to be different when you don't use cross-linking in that process. So that's kind of the commentary, but then you can think the general picture is that all of these sort of modalities are pointing directly into the regulatory DNA. Okay, so how do we see inside further? Well, I mentioned earlier that you can sequence these fragments, And ultimately, every one of these DNase fragments is originating from a spot in the genome where the enzyme, which is a lot smaller than we are, sees it actually is the only thing that's seeing what's really going on in the world, and it's making its little cut. But the idea then is that if we could somehow sample all those cuts incredibly deeply, then we could start to reconstruct what the enzyme is seeing. And this is basically done by cranking up the read depth and then being able to see, in this case now you sort of become DNase 1, you see what it's seeing, and you see the kind of the footprints that are left behind by regions, by proteins covering pieces of DNA where it couldn't cut. And just sort of give you some ideas. So like, I don't know, so here's a promoter. So now what we're doing here, let's say, is we're taking one of these regions right here, and we're just going to zoom way in. So if we zoom way in, that looks like that. Here's our scale, about 200 base pairs. We look in the middle, and we zoom in by 10x now. And instead of, remember, this plot right here was created by tracking a window across the genome and counting cleavages. But you can also go across to each base pair and count how many times, what's the cleavage rate of that base pair in the population of cells that you've seen. And then you can map per nucleotide DNase 1 cleavage. Now, if you have a small number of reads, that doesn't get you very far. Once you start to have a lot of reads, then you can basically follow the enzyme. It's coming along, cleaving. Suddenly it bumps into something here where it can't cleave, and it keeps going along like this. In this particular case, a little peak of conservation, and parked in the middle of this is a recognition sequence for a particular factor, which we actually know is occupying there. And the entire ability to recognize this is dependent on local sequencing depth. There's no mystery to it. It's purely a depth-dependent phenomenon. So you can see this, kind of over on the right. So here's, let's say, some other region. Here's recognition sequences for 3 factors. And then I'm showing what the per nucleotide cleavage looks like after you've sequenced, 25 million uniquely mapping reads, 50, 75, et cetera. And what you can see clearly is as you start getting more and more information, you start to be able to work out what's in there. And what I'm not going to go into, although you can ask me the question later if you want, is that, but I'll give you the gist of it, is that it turns out that the depth of this footprint here is actually related to it's basically a direct readout of the occupancy, the average occupancy of that factor on that piece of DNA, and it's also directly related to the affinity of that piece as well. So what this means then is if you have the depth, and you can go to regions, so it turns out promoters conveniently typically have a lot of depth. There are a lot of distal elements that have a lot of depth, and again, it's all kind of depth dependent. At 500 million reads, you'll be mapping around, well, I don't know, it's actually around 300 million reads, you'll be mapping somewhere between 1 million and 1.5 million of these footprints on the genome. But just in terms of the basic behavior and the kinds of analysis this enables, this enables now you to go and, let's say, pull up things in different cell types and then watch what's the behavior over particular recognition sequences. And so, here, for example, is a repressor of this gene. It's kind of, here it's bound because the DNase can't get at it. It's bound. Here it actually goes away. So now the enzyme can get in there. When it goes away, this gene turns on. And this is kind of, the little, and there's also some other factors that now, when this goes away, these guys now become highly occupying. So this is kind of, the basic logic of this promoter, which just happens to be a gene that is it's a nerve growth factor, and it is repressed in non-neuronal cell types by this repressor. So this is sort of the basic nature of the data, and we're going to come back to this in a little bit. The other thing is, once you start to get this kind of data at this depth, now if you think about it, okay, if each one of these things is per nucleotide DNase 1 cleavage, and here this is just truncated at about 20 or so, just but that means, think about it, every single one of those cleavages has a piece of, has a, let's say, 36 base pair stretch of DNA that's been sequenced by the sequencer, okay? So that means that the read depth coverage in these regions of DNase hypersensitive sites is extremely high, okay? And even in the garden variety sites that are out there, you get quite high coverage. And so doing one of these experiments and sequencing, 100 million or so reads, you're pretty much resequencing all of the sites, not all, but a large fraction of the sites in the genome. You're getting sequence information. And what that means then is that you can also look at the impact of SNPs. And so you can see, for example, if there are variants that are affecting the binding of a protein, they're going to result in, and you can sort of separate the alleles, right, because it's something you can do because you have the sequences, and if you can make the genotypes, then you can actually look at the impact of variants. So, for example, if I have a, let's say, this is a little footprint that's created by a particular protein, and this is actually looking at so there's two variants here. You can either have a T or a C, so you can either be homozygous, you can either be a heterozygote, or you can be homozygote for each one of these things. So here's like a T homozygote, and then if you look actually now, when we change this to a C homozygote, suddenly, the occupancy has been affected. Now you're getting cleavages in here as well. And then you can look at the heterozygous versions of this. So here is, you can actually take in the het cells, you can separate the reads that are coming from the T allele, looks like this, and from the C allele, looks like that. So, so now you imagine that if you have enough depth, and if you have enough cell types, enough variation, you can put all this information together to reconstruct at a fairly large level variants that are out there that are affecting the occupancy of proteins to different degrees. And that's, yeah.
Do you distinguish in your chip analysis? I mean, your consensus site looks very convincing, and I know the mutation when you go and you really zoom in. But have you done any, when you do the chip analysis, have you done any proteinase digest just to make sure that it's actually that specific transcription factor that is responsible for the binding? Because as you said, you're not only getting the direct occupancy, but you are also going to get the proximal sites. And some transcription factors both bind DNA as well as contribute to regulation independent of DNA. So when you were showing your chip data, do you distinguish between which?
Well, no, I mean, those chip data that's just standard chip-seq, right, or standard X-chip, right? So you're cross-linking and then going. The picture looks quite different if you don't cross-link. That's the other thing, is that a lot of the signal in some regions changes, and you shift more towards the sites that are directly bound versus the indirect sites. Yeah?
I realize it's a cartoon, but do you have a way you're showing 3 binding events on the top and 1 on the bottom and 3 like in this on the left-hand side of this?
Yeah.
Is there a way to sort of rigorously estimate occupancy from the count data?
Okay, so you're talking about absolute occupancy.
Or at least relative to, like the entire set of binding sites.
Yeah, okay, so you can certainly create some type of a rank ordering that can give you that, that perspective. We know that, for example, in the case of a transcription factor where you know the recognition sequence, in that case, and you look at the sort of aggregated behavior, not in the case of variants, but just in the case of stuff on the genome, you know that the depth of that footprint at each individual spot. Let's say it has 30,000 spots in the genome. I go to all of those things. I measure the depth of footprint and I line them all up. Okay, so you have some range, and we know that that range actually tracks really well with the kind of the in vivo affinity of the protein. But in this case, what we're assuming is that the factor is just, and let's say that's your entire sort of spectrum, right? And what we're assuming is that the variant is taking something that maybe was up here and then flipping it down, down lower. So in other words, what you could do is you would compare it to the entire spectrum of what you might call the wild type binding to get some indication. Now, there is a way to measure absolute occupancy. And this is an experiment that has been kicked around, and it's really even based on some experiments that were done back in the 80s. But it turns out to be incredibly difficult to do, the molecular biology. But I have somebody who's actually attempting it. So, but there, I mean, ultimately, it's just there's relative things that you can get a lot of mileage out of. But it is in some way nice to know exactly you know, when I see a peak height or whatever, a number of cleavages of this corresponds to this many templates in a population of cells. And, you know, so maybe that kind of information may come out. So, what I want to do, actually, speaking about looking at things across many different instances, is I want to digress just for a minute, because it seems that there is a little bit of confusion that has arisen in the community and some of the literature about what actually is a DNase I footprint, versus something we're going to call an aggregate cleavage profile. So a DNase I footprint is very simple. It's just this spot on the genome, this nucleotide here to this nucleotide there, that there's something sitting there that DNase can't cut, and that creates a footprint. Okay, that's pretty simple, trivial. That's the original definition from Gallis and Schmitz in 1978, and that's the way everybody has understood it. But more recently, what has happened is that, so imagine I have this transcription factor here, okay? And I go, and this is just one of its recognition sites in the genome. Now imagine I went to every DNase hypersensitive site in the genome, and I looked for recognition sequences of this factor in all of those sites. Okay, so I would have thousands potentially, and each of those instances would either be occupied or not occupied to some degree. Okay, now imagine I did that, and I went to each one of those, and I just collected the DNase I cleavage information around that recognition site, and then I just plotted it. And in the form of kind of a heat map in which the rows are going to be individual genomic positions, and then the rest of the heat map is just showing the cleavage around that. If you do that, you get a picture that looks something like this, and this is done for another factor for, it looks like AP1 there. And so here is, like in regulatory T cells, there's about 2,500 different instances of this recognition sequence in DNase hypersensitive sites, and each one looked at the cleavage around them and just ranked them by the, effectively by the ratio of cleavages outside the recognition sequence to that within. Okay, and that's what this sort of ranking is here. And so here, and now, and this is these white dots are just, because you remember there's 2,000 rows here, that's just indicating the degree of cleavage, and black is no cleavage. Okay, so and so now what this gives you is a spectrum. Now remember, this whole thing is instantiated on the recognition sequence. So what this is giving you is a spectrum of essentially the most highly occupied sites down to recognition sequences that are not occupied at that point. Now, you look at this though, and you say, well, gee, I see little sort of lines and things around there. So what you're seeing there is actually the intrinsic, so DNase I is not completely sequence agnostic. It has little cleavage preferences as well. And so actually with Harman, we had analyzed this, and Harman's fellow, Ellen Lazarevich, had done a really, really nice analysis of this in sort of mapping out all of the cleavage rates of all of the different, so DNase I recognizes about a 6 base pair chunk of DNA, and then you can compute the cleavage rates for each of those. So what you do is we did this experiment where we basically just took naked DNA, digested it with DNase I, and then just sequenced the bejesus out of it, and then you can basically determine the cleavage rates for all of these little hexamers. And that cleavage rate, those cleavage rates vary quite widely. If you took the highest cleavage rate and compared to the lowest cleavage rate, there's like a 1,000-fold or something difference, between those. But most of them are actually compressed in a smaller range. And you think, wow, that's kind of a lot of, might be a lot of variability. But in reality, what happens is that there's no relationship. Like the genome didn't care what DNA saw when it was made. And so therefore, there's not really any relationship between the genome sequence and these cleavage preferences, and so that they kind of bounce all over the place. And so if you think about the enzyme and looking at, let's say it recognizes a 6 base pair piece, well, let's say it recognizes a 6 base pair piece here that has this cleavage rate associated with it. Well, you step 1 base over, and now it's 5 of those plus 1 more, but that's a new sequence, and now that one may have a completely different cleavage rate. So in order to get any kind of meaningful signal out of that, that's related to the intrinsic cleavages. It means that you just randomly happen to have a spot in the genome that has a series of overlapping hexamers that have roughly the same, low or high cleavage rates. Like, let's say if you're going to create a false positive footprint, you'd have to have a situation where you had these series of overlapping hexamers that all had lower than average cleavage rates. And it turns out that that basically doesn't appear to occur with any great frequency. But what you're seeing here, so again, this is a spectrum of the highly occupied stuff to the unoccupied stuff. And in the unoccupied stuff, you start to see these intrinsic cleavages. And then what you can actually do is you can calculate in each case what is the sort of expected versus observed or observed over expected cleavage rates. And what I've shown in the side here is where DNase I footprints are called by the algorithm that I mentioned earlier, which calls around 1 million footprints and after a couple 100 million reads. And so these blue ticks across here is just like yes, no, there was a footprint called. So you can see pretty much all the footprints that are being called are up in this zone right here. And then what you can do is you can say, well, gee, what if I just took all of these guys up here, the 20% most occupied, and I looked at the cleavage, the aggregated cleavage rates around the motif. So the motif here is in the middle in this gray zone. And you can see, it goes down like that. And then I can look at the intrinsic cleavages, looks like that. And then you basically take the deviation from intrinsic, and you can see that, gee, yes, of course it's suppressing. So what the protein is actually doing from DNase I's perspective is that it is modulating, its binding is modulating the intrinsic cleavage preferences of the enzyme, which again are big if you look at the whole spectrum, but at any given point in the genome, fairly small. But nonetheless, they're being dominated by the binding of the protein. But if you look at this stuff down here, okay, this is what it looks like with the DNase I cleavage. This is what the intrinsic cleavage, and you look at the ratio. There's not really much change there. So again, these are sort of things to keep in mind in terms of what you're looking at. Now, unfortunately, somehow it has entered into the literature that people are calling these things right here DNase I footprints, okay? But in fact, this is not. This is an aggregated DNase I cleavage profile. It's aggregating information across, in this case, maybe 500 or so different sites. And so that's an aggregated cleavage profile, not an individual footprint. Okay, so back from that digression. So we talked about the fact that about regulatory regions. Let's see, we've probably skipped over this already. The distant genes. We went through that. Okay, so here's a property which we kind of stumbled on last year. Well, a couple of years ago. The paper came out last year, which has to do with what these elements are doing. So the question was raised earlier. Gee, are all these elements, you see them, are they active at this time, et cetera? And so the answer is we don't know. Well, there was this experiment that was done a long time ago by Harold Weintraub, it turns out, and Mark Rudin, and this is published in Cell in 1982, and it was a very famous experiment when it was done. And the way the experiment worked was this, is that these guys had stumbled upon, this is back just within a year or two of the discovery of DNase hypersensitive sites, they stumbled upon this site that appeared to be conditionally active. So they took cells and they would treat the cells with something, and the site popped on, and they could see it there with their southern blots. And then they raised this question: what happens if we treat the cell with this stuff, and then we take it away? Does the site go away? And if it's so, when? In other words, how stable is it in the cell? And so they did this clever experiment where they basically took a population of cells and then they treated them with something. It turned out to be a temperature stable or temperature labile virus. And then what they could do is they could then split it into two groups where they were inactivating this thing, and then other ones where they sort of kept it on. And the remarkable thing was that you could basically actuate one of these DNase hypersensitive sites, take away the stimulus that caused it to be actuated, and then they followed the cells in their experiment for 20 doublings, and the thing was still there. Now, 20 doublings may not seem like a lot, but that's actually half of all of development, right? Like from zygote to U is 44 doublings. So we're talking about like something astonishingly stable that was present after the stimulus was there. They had a follow-up paper the next year showing it with a different entity, and then that field terminated, essentially. But you know, we sort of stumbled backward into it by doing the following analysis. So if you go back to this little cartoon that I showed you earlier, and what you can do is you notice that there is this kind of sort of on-off behavior, right? Off, on, et cetera. And if you treat all of the DNase hypersensitive sites just as being present or absent, not worrying about the peak height for the moment, and you do that, and then you just take a whole bunch of different cell types. And for this, what we did was we were very careful to select cell types that we knew came from adults, that were, in other words, supposedly fully differentiated in vivo. Okay? And if you now you just take that information, so like a vector for each of these cell types, and you just cluster them. If you do this, you get this tree, and so you know here's all the cell types. Don't worry about what they are for the moment. You can tree, and you can sort of step back and say, all right. Well, firstly, whenever you see a tree, the first thing I always ask, who's on top? Okay, so who's on top? We did all these definitive cell types. We actually also threw in embryonic stem cells. So the thing on top here is ES cells. But then you can also see that this is organized into some big sort of branches and sub-branches within here. And it turns out that everybody here in this top big branch are cells that are derived from the embryonic mesoderm, and these ones here from ectoderm and endoderm. And now, we're mostly made of mesoderm, right? And most of the stuff we know about is mesoderm. And actually, then if you look within the mesoderm, it turns out that these guys here are actually part of what's called the primitive mesoderm, and then these are all the derivatives of the paraxial mesoderm, and this is derivatives of a cell which pops out of the dorsal aorta very early on in development called the hemangioblast. And what the hemangioblast does is it gives rise to the blood and also to the blood vessels, the endothelia. Okay? Which in the fully differentiated organism, right, like what does your artery have to do with the blood that's flowing through it? Necessarily you're thinking nothing, and they look completely different, but they have the same embryological origin. And then within the blood, of course, there's the myeloid cells and the lymphoid cells, and then the B cells, T cells, et cetera. Okay? So just stepping back, all of this, now this event right here occurs very early on in development, and all of these partitions occur very early on. And even within hematopoietic differentiation, these are relatively early partitions. And yet, all of this information was somehow encoded simply in the on-off pattern of the DNase hypersensitive sites of these cell types in their fully differentiated adult state. Okay? So what that does is it suggests that those cells are retaining some type, in the pattern of hypersensitive sites, they're retaining some type of lineage memory. Because otherwise, how would you be able to reconstruct this information just by looking at the fully differentiated state? And this is not, and when I say that these are on-off, you're thinking, well, is this something you can actually see in a genome browser? And it turns out you are. So like if I went and then reordered tracks in a genome browser by this grouping here, you can actually sort of trivially see that, gee, there are regions, in other words, not this, but this is also kind of regionalized along the genome, and that these patterns really are something that you can sort of detect by eye.
Do the gene expression profiles cluster the same?
Aha, this is a good question. So it turns out if you do this with gene expression, you do not get this, okay? And in particular, if you do gene expression, you will get certain groupings of cells that have similar expression profiles. But back from that, and you don't even get this relation, you get something, you can get something quasi, like the endothelia might group together in some of the blood or whatever. But all of this information is not there, okay? So none of the lineages that you go backwards from gene expression drive. And so this kind of makes sense because gene expression is all about the here and now, right? It's what the cell's doing now. Whereas somehow in this pattern of the hypersensitive sites, there is information about what happened previously that is being pulled along. And so, yeah.
Right. Is it possible that any given differentiated cell line is built upon previous, ancestor cell population? So it's really just you kind of at each layer of regulation here, each cell, you're adding in a layer of regulation, more enhancers.
Path dependency.
Well, it's not more. So under that idea, you would have a small set of elements in the beginning. They would just grow, grow, grow. Okay, but as I'm going to show you in a minute, it actually is the reverse. And so it's, but biologically though, that something like that is happening, right? So biologically, you've got a cell that is moving down, or it's moving along a particular pathway, okay? And we know that that cell is continuously, there are various stimuli, whatever, and you know, it's eventually getting down to its place, okay? And this is kind of like the famous, you know, landscape of Conrad Waddington, you know, with the ball rolling down the hill. And that landscape has several different components. It has a component of, we'll call differentiation, and that's the ball moving down the hill. It has a component of commitment, which is the cell choosing a particular valley to go into, okay? And during natural development, those two are coupled, so that differentiation forward, choosing the path, those are coupled events, and they're not decoupled. So basically, and what, you know, what Waddington, this idea came up with, which now there's a tremendous amount of support for, is that this process is super highly canalized. In other words, that the cell, and that was his term for it, meaning that there could be all kinds of perturbations to the cell once it gets inside the little valley, but it tends to want to stay, you know, there and kind of finish things out. Okay. So, but what Waddington observed, and or sort of came to the conclusion after thinking about this for a long time, and he was an observationist. I mean, he did lots and lots of, he spent, you know, hundreds of thousands of hours looking at embryos and things like that, is he basically said, you know, I just cannot think of any way in which this could happen unless there was something keeping track of the prior cell states. So he basically said, you know, the development is a historical process, in the sense that cells must be accumulating this information. Otherwise, they would wander. There's no way to think about how they can remain, you know, canalized without having, or committed, without having known that some decision was, you know, was made. So that's the sort of the intellectual framework. Harman, you were going to?
So, one interesting point, maybe it's a good point to wrap up for like a short break. You know, maybe go to the next section or something.
Sure.
My scientific part is like, I guess, where the memory would reside, right? This, the fact that you have this pattern doesn't mean that the locus where memory is stored is actually those loci, right? It could be trans-second factor concentrations, and then somehow downstream of that you get all these patterns that kind of get locked in, right?
Right. Well,
Actual memory reside.
okay. So what you are talking about is something which I'll at least comment on. But what this comes down to is that all of this landscapes we see are ultimately the readout of some concerted action of a group of transcription factors, okay? So you can't have change in the landscape that's not accompanied by change in that factor pool. But what this comes down to, I think, is a different question. You stare at this picture of Waddington's landscape and you see the ball and the commitment or whatever. But has anybody ever raised the question of what's the gravity? Why is the ball rolling downhill? And how is it rolling? And I think what it comes down to is that the gravity is essentially a specific sequence of changes in that transcription factor, you know, abundance, concentration, or landscape. That's kind of hardwired and programmed in. That it's like a developmental guide, okay? And then everything else is kind of anchored around this. And I'll get back to this when we talk about a little bit about the mouse. But I think that's basically what it is. Yeah.
Did you do this with applying IPS?
Oh, I can tell you some interesting things here. Yeah. So we have done actually DNase-1 profiling of a whole bunch of different ES lines, a whole bunch of IPS lines, and we've also done these new cells that have come out, the naive cells like ELF1, et cetera. And I can tell you that the gist of it is that if you threw all of those into this clustering, what would happen is that ELF1 would be on top, next down are all the standard ES lines, and way over here are the IPS lines, all of them, okay? Like we've never seen any IPS line that seems to be back at the level of the ES, or certainly not the alpha line cells. So, yeah.
I have a question on the first question. So if you were to cluster the regulatory patterns, you get some.
Which patterns?
The first question. So
Yeah.
if you cluster the regulatory patterns, you get 1, 8.
Yeah.
And you cluster the expression patterns, you get 0.
Yeah.
Would that mean that there are some features that are not
I think that, I think the issue is that you get a tree out of the expression data, but that tree defies any kind of explanation in terms of its relationship to development. Now, there are many things, obviously, that contribute to the effectively what we're talking about is the RNA abundance, right, when you do one of these experiments. That could be post-translation, I mean, there could be post-transcriptional, modifications or whatever. But the bottom line is, I mean, it's very difficult to make any sense out of the trees that you get when you cluster the RNA data back beyond about one branch. Yeah.
Is the genes expressed that are imparting the DNase profile you see?
Not all of them, just a few. Right? Because it's only a small number of the transcription factors, and they may be just perturbations in levels. And so even if you cluster the transcription factor genes, you still can't recover the tree.
But it's been demonstrated that you can, via use of different induced expression of different transcription factors, that you can actually roll the ball back up the hill, so to speak.
Well, what it's been demonstrated is that you do something to a cell, and you put it in a black box for 2 weeks, and at the other end, out pops a cell that you claim is at the top of the hill, right? And whether that is actually rolling back up the hill or whether that is actually entering some kind of wormhole in the space-time continuum and popping up there is a completely different story, right? Because in other words, that people like to invoke that analogy of dedifferentiation, but dedifferentiation means that you are unwinding those commitment and differentiation events. And there's really no evidence that that occurs.
What creates the commitment? What's the gauge?
What's the mechanism of commitment? That's a question for Stockholm, not for nobody knows, right? So, yeah.
There was this reported histone retention in sperm.
Yeah.
So I don't know if you can add that to the data, but does it seem to feed into the embryonic stem cell state, or is it its own type of?
So just to be clear, we are talking here about everything is somatic, right? So it's after sort of post-fertilization development. Germ cells get split off quite early into their own little world, and then things happen to them that we don't know about because there's so few of them. And then, finally they'll show up in the gametes, right, that you can actually profile, like in sperm and things like that. And there what you're talking about is a type of, so here what we're talking about is a phenomenon of information propagation that is mitotically inherited, okay? When you're talking about things that are eventually being meiotically inherited, and that's a whole different, whole different ball game.
I guess the claim with the sperm study was that this was some mechanism for conveying information transgenerationally, and that it sort of predicted or presaged the embryonic stem cell patterns of at least certain histone types. And so I was just curious.
Yeah, I mean, the thing I would say then is that is whatever that process is, it's operating way up here, before development and differentiation starts. And the thing I think with all these transgenerational, data, and there are more and more coming out of better quality, much more believable. I think that now, people used to think it was all voodoo, and now there's a lot of, good experiments that are coming out behind it. Is that that clearly some type of some mechanisms do exist, right? And for doing this, I mean, I personally believe that there are, I've seen some data, some really compelling data from mice that suggest that can be passed forward. I don't know about the specific mechanism with the, how it's getting there, but at least you can have pretty compelling evidence that some type of event or let's say metabolic insult to, the parent is passed on to certain things in the pup, okay, without wondering what the mechanism is. But the issue about it is that in every case that people have documented these phenomenology, they kind of get diluted after a couple generations. Like, it's not like a permanent evolutionary stain that's happened. So it's not even happening on a time scale on which it would impact selection kind of as we know it, And so it's a little difficult to understand what, I think that the gist of those mechanisms, if you ask my personal opinion, is that they are things for sort of protecting the immediate, F1 generation or whatever from adapting it to a particular environment, rather than sort of a generic mechanism by which the organism arises. What we got? Yeah.
Do you have any general estimation on how many DNA binding proteins will not lead to a DNase 1 segment?
Oh, how many proteins are there per hypersensitive site? It's on the order, it's less than 10, typically, almost always, yeah, except for big sites like promoters.
Because I guess for
No, this is largely due to the fact that ChIP-seq is on average measuring, you know, most of those are indirect events. So they're not the direct binding events.
Comes from the
Yeah.
I think there are 2 or 3 factors that distinctly don't have. I think the math K and math F, those 2 partners, at least from what I've seen, they're completely different body of factors. And there could be a class of factors that just don't have accessible footprints or sites.
Well, I don't know. I mean, I would say we can look at the data for those, but I think that every factor, the only ones that we have looked at that don't have, there are some of the factors that are crab zinc fingers that have this kind of repressive domain that somehow attenuates the, you know, the accessibility to the template. In other words, that they're probably binding on something that's not, you know, that's not binding. But in every other event where we've seen something binding on the DNA, you know, it's got to be there, right? I mean, it just, you know, you're, yeah. No, I mean, yeah.
How concerned are you about the fact that a bunch of these things cluster also by like what they are in the long run? Like everything that the thoracic and invertebrates are those are all fibroblasts.
They're not quite. I mean, there are a lot of fibroblasts. There's also epithelia in here, right? But there are.
Well, I mean, right. So there's some splitting between the epithelia, but a lot of the other ones, like all the endothelial cells are together, all the lymphatic cells are together.
Exactly.
But that could just be that's because their end state is so similar, not because the developmental path that led to them.
Okay. So I see what you're saying. So, but if that were true, right, then there would be a situation where, so you're saying it's just kind of converging on, well, but the thing about it is, then we're not talking about the end state here, okay? What we're talking about are the inferences about development are coming from these relationships right here, okay? So look, if you cluster by gene expression, you get these blobs also, right? All the endothelia come here or whatever. But it's these, you don't get exactly this, but you get something, you know, like I said, these blobs will hold together. But it's this information that's not there, okay? So, and that's what you're talking about. So this is the stuff, this is about the actual active state, right? And this is about how they ended up. But that doesn't, there's no necessary relationship between this and, for example, this partition right here.
So in terms of cause and effect, you're saying that it's the pattern of these activation regulatory elements that are potentiating the development for each of these lineages and maintaining it?
Well, no, I'm saying that what we're looking at here, members, we're looking at the shadows that are being projected on the wall from the real events, okay? And the real events are ultimately, you know, that transcription factor kind of cascade that I was talking about to answer Harmon's question, right? So the DNase landscape is just a readout of what's going on at that level, right? And there's something controlling that, which is controlling the forward movement here of development. And there's ample evidence, obviously, for the causation of transcription factors in, you know, in development.
But aren't you, I mean, back to Armin's point, aren't you then ignoring so much that happens post-transcription?
Well, I guess you would say the following thing. If I can derive all this information from looking at those events, then, you know, I mean, there are lots of phenomenology in cells and biology, right? It's just a matter of ranking of importance. And so, you know, so, yeah, I mean, it's just, I would say this. This is more like, this is just, here's the phenomenology, and then, but there's a bunch of other interesting points that can come out of this. We'll take one more question, then good. 2 more, okay, go for it.
I'm just a little bit, I guess, shocked by the result that you talked about iPS cells, because they do seem to have a very stable phenotype. They maintain their stemness, but perhaps they're retaining epigenetic memory of where they come from and don't really open up maybe chromatin sites that are present in the AS cells, but not present in the iPS cells?
I'd say our analysis of the IPS line shows 2 things. Number one is what you alluded to, is that they retain an imprint of the cell type that they started with. And then the second thing is that they just do not seem to make it all the way back. I mean, that there are pretty large sets of things that distinguish ES cells from other cells that these cells don't have. So that doesn't mean that they can't be themselves some kind of a stable cell, right? I mean, stable in the system. But you got to also have to realize that ES cells, IPS cells, whatever, these are completely unnatural phenomena, right? Because all natural cells, nothing ever sits still in nature except for the terminally differentiated cell. It's always differentiation, always forward moving. There's nobody, the only example we know of things stopping is when something peels off to the side, like a hematopoietic stem cell that will renew itself and then also kick off progeny going down. So these things are already kind of outside of the regular framework of nature. And you know, when you have those IPS cells, so let's think about this as well. So remember, in normal differentiation, in normal development, differentiation and commitment are coupled. We know that you can take IPS cells and differentiate them, but there's no evidence that those are good for commitment. Right? There's no evidence that that cell put in the, you know, is not going to wander or something like that. So there's a lot of questions surrounding that state. So maybe it's possible to get commitment and differentiation starting from that state. Maybe you have to start at a higher state. Who knows? But I think it's just mainly being aware of what the potential, you know, issues are, but that they're clearly not the same as ES cells, I guess. And I think probably everybody would agree with that.
Okay,
Yeah.
this is a great
Break
time
time.
to take a 10-minute break.